Let's move on to the next oral presentation entitled Effect of Mitochondrial Sodium-Calcium Exchange Protein on Radiosensitivity of Lung Cancer, presented by Ms. Ziyue Zhang.
I will share my screen. Can you see the screen? And can you hear me? Dear teachers and students, I'm Ziyue Zhang from Naval Medical University. It's my honor to give my presentation here. The topic of our group is the effect of mitochondrial sodium-calcium exchange protein, NCLX, on the radiosensitivity of lung cancer. Among all types of cancers, lung cancer has the farthest increasing incidence and mortality rate, seriously affecting people's health and life. Therefore, the effective treatment of lung cancer and the extension of the survival rate of lung cancer patients is one of our important research objectives. In the treatment of lung cancer, radiotherapy is an important way to treat lung cancer. Radiotherapy makes cells produce a large number of ROAs, destroy DNA and other biological molecules, causing tumor cells to die. Therefore, taking ROS regulation as the core has become the most direct research idea to enhance the radiosensitivity of tumor cells and overcome their radiation resistance. The current regulation of ROS is mainly carried out through the above 4 channels, but it didn't work out well. So it is very important to find new targets to enhance the sensitivity of lung cancer radiotherapy. The important role of NCLX in reactive oxygen species was first reported in Nature in 2020. After irradiation of tumor cells, ROS produced by mitochondria, but ROS decreased after the addition of NCLX. Imagine using NCLX inhibitor to increase ROS, which will increase the damage of tumor cells. It is worth noting that the specific mechanism of NCLX has just little research, and its role in radiation damage has not been reported. Therefore, our group supposed, can NCLX be used as a new target to enhance radiosensitivity of lung cancer? To test this hypothesis, we developed such a research idea. First, in animal experiments, we evaluated the radiosensitivity of lung cancer and its effects on normal tissues. Second, in cell experiments, we explored mouse lung cancer cell LLC about its radiosensitivity and the effects on ROS pathway and apoptosis pathway. Finally, investigate NCLX affects DNA damage and repair by regulating ROS. We did cytological verification of the cancer cells before the formal experiment. In flow cytometry, there was no obvious apoptosis of cancer cells in the control group and the single inhibitor group, but the apoptosis rate of cancer cells increased significantly in the irradiation plus inhibitor group. EDU proliferation assay showed DNA proliferation in the irradiation plus inhibitor group is reduced. Based on this experiment, our team verified that NCLX inhibitor has enhanced the radiosensitivity of lung cancer. After that, the team began to build mouse lung cancer radiotherapy model. The model has the characteristics of difficult operation and low tumor formation rates. We will add NCLX inhibitor to the tumor-bearing mice and irradiate them, which will be the basis for subsequent experiments. This is the lung tissue with successful tumor bearing. It can be seen that there is tumor formation on the right lung tissue, and at the same time, the left lung tissue has also sites and a large number of infusion can be observed when we opened the chest cavity. Internal assay showed apoptosis was evident in the tumor tissue in the irradiation plus inhibitor group. Ki67 assay showed that cell proliferation was inhibited to the same group. These two experiments confirmed that NCLX inhibitor has the effect of enhancing radiation sensitivity on cancer cells after radiotherapy in animal experiments. The above are the main experimental results of our team so far. Next, we will report the experimental arrangement of our team in the later stage. Firstly, the tumor-bearing lung tissue of mice was pathologically detected to observe its sensitivity to radiotherapy. Subsequently, the vitality, proliferation, apoptosis, and circle were detected by CCK8 to explore the sensitivity of cancer cells to radiation. Finally, by detecting ROX content and NCLX expression, the influence of tumor cells inhibited by NCLX on ROX pathway and apoptosis pathway was explored. In the mechanism exploration, we will verify the friendship between NCLX and ROX regulation to clarify the relationship between NCLX and DNA damage and repair. Through a large number of experiments in the early stage, we felt that we should have enough patience and tenacious perseverance and work unite to tide over the challenge. That's all. Thanks for your listening.
Comments or any questions?
Thank you so much.
Thank you. Thank you.